here. I'd like to invite you to the next meetings of the three associations as well. The ISTA annual meeting will be held next May 29 to June 1 in Verona in Italy. The location will be the Hotel Leon d'Oro. And the next one is the ISSS 14th Biennial Conference in July 3 to 7 next year, to be held in Paris, France at the Sorbonne University. And last not least, I would like to invite you also to the SER 2023 10th World Conference on Ecological Restoration, to be held from September 26 to 30 in Darwin, Australia. We have a new member here. Normally it's an ISTA-ISSS webinar, but I would like you to introduce our guest today from the Society of Ecological Restoration, the International Network on Seed-Based Restoration. Their past chair, Kingsley Dixon, will introduce in a few minutes this association. And Kingsley is a restoration ecologist and conservation biologist, professor and director of the Center of Mine Site Restoration in Western Australia, with about 30 years experience in restoration focused on mine site and urban interface restoration and conservation practices. What I found as well, Kingsley, you are Western Australia Scientist of the Year 2016, and I would like you to take over. Therefore, I need to make you the presenter. No, who's presenting you?
Simon's presenting.
Yeah, I'll get the slides when Kingsley talks on it.
Thank you. So while you get that up, Simon, if that's okay, I'll talk. So thank you for this great opportunity where a key section of the Society for Ecological Restoration, the International Network for Seed-Based Restoration, is able to present one of our key initiatives. And SER is the peak global body for ecological restoration. We have 4,600 members, over 110 countries globally, and growing rapidly. I'm the current chair for the society. We're based in Washington, DC, but we have chapters in an amazing array of countries, including Australasia, which is the Pacific Basin. The INSR is one of the biggest of the sections, with almost 600 members and growing rapidly. And I think that reflects the growing need for seed and seed-based restoration. And this evening, today, this morning, it's a very special occasion. Next slide, please. Because I'm really thrilled to launch the new SERQ Native Seed Information Database, otherwise known as SID, which has been a global resource developed under the Millennium Seed Bank, which is now being relaunched with an SERQ co-branding, and that's being homed through SER. And I acknowledge, if you press next, please, acknowledge the great support, Simon, if you, yeah, for the Australian Centre for Mine Site Restoration at Curtin University, who sponsored the transmission and translation of that information to SID. Why is SID important? It is the key global database for native seed information, with 185,000 seed records and growing daily. We encourage all members who work in native seed who are on this call or listening to the recording to please log on. It's simple, it's straightforward, and you're going to see how to do that shortly. I'll conclude with a short 3-minute Minute Earth. It was launched on Monday at COP15. you can see it under Google. If you just Google trees won't save us. We're going to play this in just a minute. Simon, if you just hold off for a minute. This was launched on Monday at COP15 in Montreal. In an extraordinary feat, we now have reached almost 280,000 views in just 3 days, likely to hit 1 million by the end of the year. So without further ado, I will pass over to Simon to run this Minute Earth, which really tells you why getting smart with seed and being part of the Native Seed Global Network is so important as we move towards a renewed way of managing the planet. Thank you, Simon. We need the audio. Do people get audio or not?
There's no audio.
No, Simon, if you could rewind, please, just to play the audio. You're gone on mute somehow.
Okay, now it should work.
Okay, thanks. No, not working. Have you put the volume up on the YouTube channel? It's down in the right, bottom right. Oh, what a pity.
Maybe, Kingsley, you have to sing and guide us through it.
Sorry. Apologies. It's not working. We might move on to Simon, I think, at this stage. It's a great pity, everybody, because it's not working. No.
Okay.
Thank you all. Okay, over to you, Simon.
What you can do, you can get everybody in the audience the link, and everybody can have a look at it at a later stage. Thank you very much for the introduction into SER. We would like to start now with a short presentation of about 30 minutes from Simone Pedrini. What I would kindly ask you all is, if you have questions in the audience, please put them in the question and answer box. And if we have a few minutes left at the end of that presentation, we will allow questions and we'll pass these questions which you put through in the question and answer box. Let me introduce Simone. He's a lecturer in environmental restoration at the School of Molecular and Life Science at Curtin University in Perth, Western Australia. He specialized in native plant species conservation and restoration, and his main focus is on optimizing the nature seed supply chain for ecological restoration. He's got a PhD in seed enhanced technology application to native seeds from Curtin University, and Simone is certified ecological restoration practitioner, chair elect of the International Network of Seed-Based Restoration, and co-founder and advisor of the European Native Seed Producers Association. Simone, the floor is yours for the presentation. Thank you very much.
Okay, thank you, Andreas. Checking that everything works fine. Can you see my slides?
It looks good. Thank you.
Excellent. Thank you very much. So, as already introduced by Kingsley, we specialized the international network for seed-based restoration into looking for solution to some of the key issues in ecological restoration when it comes to the use of native seeds. Before entering the details about the native seed supply chain, I want to give a brief introduction on what can be considered acceptable native seed material for restoration. In this definition, a seed batch is to consider appropriate for restoration when it has a certain degree of genetic diversity, which has to be representative of a population where this material was collected in the wild. And this genetic diversity needs to be preserved and maintained throughout the entire supply chain and eventually deployed at the restoration site. That means that all materials that have undergone active process of selection of improvement to turn them into varieties, also called in America as nativas, is actually not advisable for ecological restoration. And that's because in the genetic diversity and the local adaptation, seeds the capability of these seeds and those plants to survive these harsh environments and be able to evolve with the environment. And this, I think, is one of the key differences between two products that look so similar. Some is the varieties of horticultural and crop species, and some of the seeds of natives. So for selected and improved variety, they usually derive from long periods of plant breeding and selection, and that's done to select some beneficial traits like synchronous seed maturation, or seed shattering, or removal of dormancy. And those traits are all akin to make them easier to handle, more productive. And so it's important to maintain the genetic purity of this line and guarantee the varietal stability. On the contrary, native seeds are representing this very broad array of genetic diversity of a natural plant population. And because of the diversity, there can be lots of interspecific variability between different populations, but sometimes even between individuals within a single population. And that can bring local logistical complexity, different seed morphologies, different dormancy patterns. Sometimes those differences can be very hard when trying to handle those seeds or when trying to test their quality. And the key information when it comes to native seed to maintain throughout the supply chain is the origin, where they came from, as long as those seeds were collected in an appropriate manner. But the most important difference between these two kinds of products is that selected and improved varieties are usually used to produce foods or goods or some service, while native seeds are used to restore ecosystem where that diversity, as I said earlier, is crucial. Unfortunately, those differences are not clearly understood by the regulators. There's a problem that we try to apply rules that were developed for selected varieties to a completely different products, with the side effect of limiting and hampering the development of those industries, as is seen, for example, in Brazil, or we can see also in Europe, with this directive that was developed 12 years ago, that has the scope to regulate the native seed market, but as a side effect is actually limiting its development in many countries. Enough of like the theoretical talk about natives. I want to show you an example of why and how can we use native seeds. And I want to bring you here where I'm currently located in the southwestern corner of Western Australia. This area has undergone a severe process of land degradation and destruction in the last 150 years. You can see the black spot expanding is the area that has been cleared from native vegetation in the last 150 years. And now it's not uncommon to see pictures like this one, where small patches of remnant bushland are surrounded by farmlands and paddocks. The problem with this approach is that now so much of this natural ecosystem has been cleared that we start losing the ecosystem services that this system provides. And that means that now there's severe problems with, for example, wind erosion or salinity that is affecting the very crop they're trying to grow there because that vegetation is no longer there to provide those services. In the last decades, they tried to mitigate this problem by reintroducing some native vegetation. And the most common approaches are the use of tree planting or direct seeding. And a very common question I'm asked, what approach shall we use, plants or seeds? And I'm going to show you a little simulation of these two scenarios. Let's say we have 1,000 hectares need to be restored to a native bushland in this area of Western Australia that is called the wheat belt. In the first example, we use seedlings. Our target is to get 2,000 plants per hectare, meaning that to cover the entire area, we would need 2 million plants. But we'll have to assume that not all of those plants will survive. So with an assumed success rate survival of 50%, we know that we will need a total of 4 million plants. So at a cost of $1 per plant, they cost $0.25 to put them in the ground, and combine all these numbers, it's going to cost us about $5 million to cover 1,000 hectares with all these plants. Let's look instead if we try to use seeds. Same target, 2 million plants as our outcome. Seeds, however, are way less successful than plants in establishment. We can assume a success rate of about 2%, meaning that just 1 out of 50 seeds will result in a mature and healthy plant. The question is usually, how many kilos of seed do we need? And that can be a very useful parameter if you're using a monoculture, for example, with canola or wheat, where the weight of the seeds is usually known. But when it comes to natives, it's much harder to understand what a kilo means, because when talking natives, you might have a mix composed of tens or hundreds of different species. And I showed this example showing 4 species that we might use in such project. One can be this very tiny seeded melaleuca called honey myrtle. Another example is an eucalyptus, or a saltbush as a Chenopodiaceae, or an acacia, a legume. As you can see, the difference in shapes are quite clear, but the difference in size is immense. For example, 5 seeds of the acacia are going to weigh as much as 1,000 seeds of the melaleuca. And that's not even taking into consideration quality. We run the viability test of some batches on this species, and you see how variable is the quality, going from 10% to 77%. So taking into account quality as well, that changes the number of pure life seeds we actually get per kilo. You see how easily it can get complicated. And now I'm showing you just 4 species. Imagine doing the same for hundreds of species. So we are suggesting to take a different approach. Instead of talking of kilos of seed, let's talk about number of pure life seeds instead. It makes life so much easier for the restoration practitioner trying to design the restoration seed mixes. So let's talk about pure life seed number, because we know that we need 2,000 plants and that just 1 out of 50 of them is going to survive. We know we need 100,000 pure life seeds per hectare. That means that in total, we're going to need 100 million pure life seed. And we know that 1,000 pure life seeds cost $6.33 Australian cents for Australian native plants. And we know that because we ran the analysis over almost 130 species, which we obtained the batches, we obtained the prices, and then we performed seed quality testing, where we could understand the 1,000 seed weight and the price, and then estimate an average of $6.34 for 1,000 pure life seed. Multiply this number by the number of pure life seeds we actually need, add the cost for delivering seeds to site using tractor and preparing the site. It gives you a total of about $800,000, which is 6 times less of what we paid for tree planting. And this is one of the key reasons why there's so much focus and interest in using native seeds, because it can be much more cost effective than tree planting. That said, it's usually better not to rely entirely just on one system. It's better to use both. Not all species work well from seed, not all species work well from seedlings. So it's better to do a bit of bet hedging and try both approaches. The key question now right here is, if you notice, especially if you come from an horticultural background, how come 2% success rate is acceptable? And also, how come it's so expensive? How can we reduce that cost? And to try to tackle those issues, it's important to understand how the native seed supply chain works. So, native seed supply chain, I like to look at it in a broader context of a restoration project. where you start from an ecosystem that needs to be restored. Through a series of restorative activities, return a healthy and restored ecosystem. First step usually is to develop a restoration plan. And within this plan, you have a native seed sourcing and procurement plan, where you decide what species you're going to reintroduce, where you're going to get the seeds, and how many seeds do you need. It's from there that the actual native seed supply chain starts. It starts from the collection from natural populations. This stage seed can either go straight to the side, or for using scale of economy of scale and increasing the quantity of seed available, they can be multiplied in seed production systems. Once collected or produced, seeds can go through seed processing to improve the quality, eventual quality testing to actually quantify and understand how viable those seeds are. If necessary, can be stored for some time. For species that present dormancy can be alleviated. If you want to get fancy, you can try to apply some seed technology to improve the success of establishment, and eventually go to side. You seed, and over time, through monitoring and adjusted management, hopefully you'll get your restored ecosystem back. All of these steps of the native seed supply chain have been discussed in depth in a series of publications that was published 2 years ago on the journal Restoration Ecology as a special issue. All of these articles are open source and freely accessible online. I've left the link in my presentation. I'm going to share the PDF of the presentation afterwards. And if you're interested, just click on the link. You've got free access to all of these publications. And the international standards for use of native seeds in restoration was produced by the International Network for Seed-Based Restoration, which, as Kingsley said earlier, is this organization that has members now in 60 countries, almost 650 members. If you have the time, I'll encourage you to go and visit our website. We tend to release, at least once a month, some news on the native seed space. We'll post events such as conference, webinar, or workshops, and also post opportunities for employment or for studying in native seed. Maybe more interesting is the webinars section, where we can have, you can see webinars, for example, a very interesting one, how native seed production is structured in Germany, which is probably one of the most sophisticated countries in the world when it comes to production of native seeds. There's another one, Kingsley Dixon, explaining the history of the discovery of smoke and its effect on seed germination. Or some curious little project, as example, building a portable after ripening bucket for relief dormancy on small seed batches. And this is just an example of the sort of material and resources that you can find on our website. The coming soon is a very large project that we've been developing in coordination with the Society of Ecological Restoration and the Bureau of Land Management in the United States, which is a documentary series on the native seed supply chain, mostly in the United States, but that truly encompass all of the principles that we explain in the standard. This series of videos is actually produced by serious and professional seed producers, so I'm very confident it's going to be an excellent product, and it's eventually going to be released for free online on YouTube and various platforms. So stay tuned. Hopefully we'll be able to release them at the end of March next year at the Native Seed Conference in Washington, DC. Okay, without further ado, I'll jump on what I think is the main reason why most people logged in to my talk, which is the presentation of the new seed information database interface. Most of you probably are familiar with this look. This is the original seed. It was a fantastic tool developed more than 20 years ago by the Royal Botanic Gardens Kew with the Millennium Seed Bank initiatives. The beauty about this system was that it was freely accessible to anybody, was fairly intuitive and easy to use, and especially had lots of data on lots of species. As Kingsley said, there's almost 55,000 species contained in this database. And so I was shocked that when I heard earlier this year that unfortunately, because of it being developed more than 20 years ago, it was running on an obsolete platform, and it was no longer compliant with some internet accessibility regulation in the UK, and therefore needed to be shut down. It was a bit of a shock to me, as I guess it was for most other seed users. And so with INSR, we started a sort of a campaign and tried to understand how impactful the closure of seed would be for the seed community. And for that reason, we launched a survey earlier last year in March, where we asked a very simple question. It was surprising that we obtained almost 240 answers in a matter of weeks. This is the kind of people that replied to this survey. It's mostly researchers, since scientists and restoration ecologists. Unsurprisingly, lots of conservation seed banks, but quite a lot of native seed suppliers and restoration practitioners as well. And a fair bit also of people from education sector. So we know that seed has also been used for education purposes. I then asked the question, how often do you use seed? And we got a variety of answers. Someone uses just a few times a year, a few times a month, and 18% of people said they use it on a weekly basis, and 7% also on a daily basis. Very important answer is about how much would seed closure affect your activity, either as a researcher or as a seed business. And more than half of the respondents say that it'll drastically affect the way they operate. I think the most surprising part about the survey, and it's a good indicator of how passionate people are about this database, is that at the end of the survey, I left an open question, a volunteer open question. Who wanted could comment on it, and more than 60% of the people actually provided a response. I filtered down 3 of the more interesting ones for you to read. The first one is from a seed bank. As a seed bank, I regularly refer to seed for species with unknown germination requirements. It is often the first resource I check. There is nothing else out there like it. Another very good one is, we are a small non-profit that both collects our own seed and grows out material for our restoration projects. We have limited capacity, so seed takes out some of the guesswork where it comes to finding out what kind of pretreatments and temperatures are optimal for germination and storage. Now, my favorite, seed is one of these resources that is freely available to seed researchers, seed users, and restoration ecologists who need to figure things out. As such, it is a good resource and should be maintained. God knows the folk doing restoration have it tough enough. So these are all clear indications that the seed community really was standing behind this database and really wanted it continued. And this eventually has escalated this discussion with Q and SCR, and we've reached this agreement that Q would pass on the database to SCR to be hosted on a separate server and run by INSR to maintain it free and easily accessible. And while doing so, we've redeveloped the interface, as the old one and the platform were obsolete. And this is what the new interface looks like. And I think the best way to show you this new interface and its functionality, I'd rather show you directly on the browser. Hopefully it'll work. Andreas, do you confirm that you can see my screen, the browser?
Yes.
Excellent. So, as you see, compared to the previous interface of Seed, this is much simpler. The only thing you can see is the search bar. And it works simply that you start typing the name of the genus of the species you're looking for, and after 4 letters, that series of all the species present in that genus will appear. For example, let's try Digitalis, and here we go. We know we've got 66 species belonging to the genus Digitalis. You can try with any genus or species. Let's try a very large one, Acacia, and we got 867. Let's go back to Digitalis. So if you see here on the left, you've got the list of the species in this genus. On the right, there are a series of icons. These icons represent what sort of information are available for that species. For example, this icon with the scale means that we have the information about the 1,000 seed weight. The box, that means there's information about storage. This little stake means there's information about protein content, yellow dot, oil content information, and the seedlings germination data. I don't think, here we go, squirrel means that there's information about seed dispersal, and another one, which is very rare, because just 500 species at the record, is seed morphology, which is shown as a ruler. Unfortunately, it's not present for any of the digitalis. Now, I want to show you the record for one of these. Let's select the foxglove, digitalis purpurea. As you see, this one's got lots of information on it. When we open it, it's how it looks like. First box, you have information about storage behavior. In this case, is orthodox. And there's a table with all the information collected about germination. So germination percentage, condition it was at, medium it was grown in, and also the seed, the provenance of those seeds as well. In this species, there are lots of germination results. There's a box about seed dispersal. We're talking about dispersal agent and the methods of dispersal. And we've got seed weight. Here also reported are all the measurements of 1,000 seed weight. On top, you also get the average of those seed weight. Similar for oil content, different measurement for oil content and the average, and the same for protein content. And at the bottom of the page is the references that those information were sourced from originally. So this is the way each species now look. Species with less data will look simpler, species with more data will look longer. It's the same, similar as it was in the original seed, hopefully a bit clearer. A new change in this system is that now we can keep track live of how many records are available. So we know exactly we've got 182,232 records from 54,083 taxa. In these boxes, you see how many records are available for each of those traits. As in the original seeds, there are pages that describe each of these traits and how they were obtained. Difference to the original seed is that we want to give more visibility to one of the fantastic tools the seeds had, which are the viability calculators. We decided to maintain 4 of them, which is actually 2 of those calculators we thought were the most useful. First one I want to show you is the equilibrium moisture content. It's a way that by knowing the oil content of your seed and by knowing the drying temperature and the relative humidity in the drying environment, you can give an estimate of what is the moisture content of the seed, which we have faster methods than drying the seeds in the oven for 17 hours. So for example, the oil content is 10%, our drying temperature is 15, and our relative humidity is 15%. That gives us a percentage of the moisture content. If you don't know the oil content, you can search along all of the species that this data are available. There's quite a few of them. So let's try with the example before. Let's try again the foxglove, digitalis purpurea, and you can select which oil content you want. Stick with the one from Fiona and calculate. We can see that because this species has got very high oil content, about 37%, moisture content at this condition is about 4%. By changing these parameters, so instead of drying it in a fairly dry environment, we'll dry at ambient condition about 50%. You see now the moisture content is almost 8%. This information is useful when you go and use the viability calculators. These are exactly the same calculator, but presented, but returns different results. For example, one is the storage time. This is a bit more complicated than the previous one because you need to enter more parameters. These parameters here can be obtained by performing some rapid aging experiment. For some species, those have already been entered. And we're lucky because we got the same example of digitalis purpurea. Let's click here, and this fits all these parameters for the equation. The equation that we use here is the Ellis-Robert equation that was developed in 1980. After entering these parameters, we can now use the equilibrium moisture content, which we've calculated earlier. If you haven't done that, you can actually open the same calculator here, select your species, for example, yet again, Digitalis purpurea, and enter your condition, 15 and 15. That returns you moisture content. You need the seed storage temperature. Let's say we'll keep it at 5 degrees. Our initial viability is 95%. Now the question is, how long will it take for those seeds to drop the viability, let's say, by 10%? So we want the final viability to be 85%. Submit. We've got the results. So this condition with this moisture content at this temperature will last in storage for 14 years, dropping 10% in viability. You can see by changing, for example, the moisture content, let's do it at 8%, submit, all of a sudden our storage time is down to 107 days. Exact same calculator also work on the other way around. So instead of asking how long will it take to drop viability by 10%, I can ask how long, what the viability will be if I keep my seeds in storage for so many days or years. Let's try it again, the example. Digitalis purpurea, equilibrium moisture content 4%, storage temperature 5 degrees, initial viability 95, and we want to store them for, say, 1,000 days. Submit, and it gives us 93%. If we change the moisture content to 8%, submit, and we see that after 1,000 days, all of the batch is probably dead. This is especially useful as a tool for teaching. So when you try to get students to understand the importance of appropriate storage condition, this is a very useful tool. Unfortunately, there's not many species available in this list. So if you're aware of studies where such information were obtained, or you're performing studying about this, let me know, because we'll be keen to include them in this list. So this is it about the new interface of SEED. As you say, is very simple. We've now gone through the process of finalizing it. There's still a few things that need to be integrated. We still have to add the logos of the various organizations in this page. Fundamentally, the transfer of the original seed is now completed. Next step in the process for us is to try and include more data to the database. We're still working on a system that allows us to insert data in the database more effectively, especially with data dumps of big databases. But hopefully in a few months, we'll be able to sort it out, and we can start taking donation of data from reputable sources to increase the data available to the community. So to conclude my talk, I'd like to acknowledge and thank the great help of the Royal Botanic Garden queue in facilitating and allowing this transfer of the database. Of course, thank you for the Society of Ecological Restoration and INSR for agreeing in taking on board the SEED database and the continuation of development of the database. As Kingsley mentioned earlier, special thanks to the Center for Mine Site Restoration and Curtin University to providing the financial support that allowed this transfer to happen. But most importantly, I'd like to thank Chid Gilovitz. Without his contribution, nothing of this would have been possible. He's an incredibly talented programmer, which also happened to be an excellent botanist. And the combination of the two make my work much easier because he understands plants and he understands programming. Special thanks also to Robert Turner from the Royal Botanic Garden queue. He was one of the original developers of SEED, and it was extremely generous and helpful in assisting us during the transfer. And also special thank someone that most of you probably know, Fiona Hay, that was incredibly helpful in redeveloping the SEED calculators and making sure that everything was working appropriately. That concludes my talk. This is my email if you want to get in touch with me. As I said earlier, I will share, or maybe Joanna can share the PDF of my presentation with you all, so you can use the link. And if there's any time, I might take a few questions.
you very much, Simon. There are a few questions, and I only take the ones, a few of them, which are which reached me before. There's one question: is there a benefit to using a combination of nativas and wild harvested genetically diverse seed?
So it depends. It depends. Like the advantage of nativas is they're probably faster and cheaper to produce. The problem is they don't have that same genetic diversity. In some scenarios, it might be appropriate to use them. For example, after a wildfire, there's not enough native seeds of appropriate origin, and you want to stop erosion, or you want to focus on a specific service, then yes, in that case, it's better than nothing. But if the native alternative is available, which is of appropriate origin, that will be the preferable choice, as I said, if it's available.
Okay, thank you very much for that. And we may have a few others. What about area species markets where seeds harvest costs are equal or greater than planting costs?
Sorry, say it again. I missed the beginning of the question.
What about the area species or markets where seed harvest costs are equal or greater than the planting cost?
Is he talking about aerial seeding?
Yes, probably.
Ah,
Yeah.
okay. Well, it all depends on the scenarios. Like on a former farmland, you wouldn't use aerial seeding; you would use just a tractor. But in certain scenarios, yes, it also dependent. And for example, for very steep mountains, you want to use hydro seeding, but that is also incredibly expensive. And that's why I say you should never use one or the other. It's usually better to try to use a combination of both. But in some scenarios, tree planting is far better. In other scenarios, you can get away just with seeding. So it's never just one or the other.
Yeah. Do you have a methodology for multiplying seed to maintain diversity? That's a very tricky one, I think.
Yes, it's just being incredibly careful. There's a methodology. Try to avoid selection whenever possible. Some degree of selection will happen. It's unavoidable because the time you decide to harvest, you already performing some degree of selection. When you clean the seed and some smaller seed go through your screens, you've already performed selection. So to some degree, it's inescapable. It will happen. There are some guidelines that, for example, to avoid too much of breeding, they suggest to do not multiply the same species for 5 generations continuously without introducing new material. That depends, because some species do self-pollination. For example, 3 generations is more than enough. Like you already start seeing some of that in breeding depression. Other species, wind pollinated, might actually go for longer. There's lots of interest in starting examining all these aspects. But as a general rule, it's all about being careful and avoid any voluntary selection, let's put it that way, and limit as far as possible all the involuntary selections.
And maybe a last one, is there a plan to have these species also in the global seed vault in Norway?
Some I think already are. I don't know to what extent or what they consider valuable enough to go in there. I've never had any of my seeds been sent to the seed vault, but I think probably there's some very rare or conservation concerned species that we had enough seeds for. Maybe they can send it there. But probably I'm not the right person to answer this question.
Okay, thank you very much. There are more questions in the question and answer box, and I will assign them to you, and maybe you can find them and try to answer them. I will collect them also and send them over to you in general. One of my colleagues will do that. I will kindly ask you to move the Kirk as a presenter now.
Yes.
To assign him. And I would like to introduce Kirk Remond. He is presently chair of the Statistic Technical Committee. He and Dr. Jean-Louis Lefort, the vice chair, they work together in very close partnership over the last 2 decades to provide statistical solutions training and tools to the ISTA technical committees and beyond that to seed testing professionals worldwide. Kirk is also a senior science fellow and statistician at Bayer Crop Science, supporting seed physiology, seed genetics, and seed health testing for the company in a global base. He obtained his degree in statistics and mathematics from Brigham Young University in the United States in the early 1990s. Kirk and his wife, Care, reside in St. Louis, Missouri, close to the Bayer location, and love spending time and leisure with their family and 3 grandchildren. Kirk, I hand over to you. You may also put on your camera that we can all see you. There's a final information as well. This session is recorded and will be during the next days available on the ISTA YouTube channel. Thank you, Kirk. Please go ahead.
Thanks for that nice introduction, Andreas. And is my screen sharing okay, sir?
Yes.
Okay, wonderful, great. Okay, well, Jean-Louis and I really appreciate this opportunity to share to this wonderful audience some of the good work being done in the International Seed Testing Association, and the Statistics Committee in collaboration with the other technical committees, and excited to share some of this, along with some statistical seed sampling or seed testing tools that are available, excuse me, on the International Seed Testing Association website. So just an overview, just these 2 main points. We'll talk about the some ISTA Statistics Committee activities, really as a basis for the development of some nice tools that we'll share with you. There's been a lot of utility within ISTA and beyond ISTA's borders to globally to seed testing professionals around the world. So here's a picture of Jean-Louis Lafond and I this last spring in the US in Johnston, Iowa, in a working meeting. We have some very engaging working meetings. We get a big whiteboard or blackboard, and we're writing formulas and making some good progress. It's exciting to us, and I think the tools at least will be exciting to you all, we hope. But Jean-Louis and I have been working together with the Statistics Committee almost 2 decades in developing these tools and solutions, and it's been our pleasure to do that, and we look forward to the future and this working in this area, both Jean-Louis and myself and our committee. So these are the types of activities that we're engaged in. Just a brief overview and we're involved with testing plans and method validation report reviews, and a lot of them associated with ISTA rules proposals. And this is just a few little pictures on the right showing a sampling of some of these outputs. Also, of course, a lot of associated statistical analysis and simulation. We find that with the improvements in technology, simulation is very useful in actually validating a lot of our calculations, making sure that they're right. So working with Fiona and Seed Science and Technology, some acting as referees for some of the papers there. I'll highlight some I have bolded here at the technical theoretical contributions that Jean-Louis and I and the committee have done, benefiting the association, but more broadly seed testing professionals everywhere. We'll share some sampling of the seed testing tools development. We do a lot of this kind of work. We also have a lot of workshops over the years for both ISTA workshops and industry workshops. Of course, a lot of collaborations within the ISTA technical committees, the executive committee, and our industry and across the seed testing industry. And then also we handle a lot of questions coming from the the technical committees and the broader ISTA membership and beyond to seed testing professionals beyond the ISTA borders. And then also looking with this ISTA initiative for Young at ISTA, the next generation of the up and coming seed professionals, seed testing professionals, really making an investment there. Jean-Louis and I and the committee are focused on this as well. We'll just give you just a brief sampling of some of the theoretical work, and we'll show a little bit of mathematics here. Don't worry. We just want to show this to you to give you some confidence of the foundational. The work that we're doing has a strong theoretical foundation. Some of the good work, still very relevant work from the 1960s, 70s from Miles and his associates. With the advance in statistical theory, Jean-Louis and I and the committee have validated some of the work with the strong theoretical basis, and this really is a nice springboard for us in the development of tools that folks can use. Here's a paper published a few years ago that's going to be a foundational paper for us for a lot of the work that we do. So this exact theoretical distributions around replicate results in a germination test. Pretty basic. We just a little cartoon here on the left. We have a 400 seed sample here, of which we have 360 normal seedlings, or about a 90% germination rate. So these can be distributed across these 4 test replicates of 100 seeds in different ways. Randomly, they can be distributed. You could have a distribution of 90 seeds, 90 normal seedlings in each of those as one possibility. Another one, 99 in one, 70 in another, 94 and then 97 normal seedlings is another possibility. There's many realizations and possibilities here. So considering this as a basis, this paper talks about the distribution of the number of normal seedlings and reps follows this thing we call a multivariate hypergeometric distribution. Don't worry about that specifically, but it is Jean-Louis and I have a strong understanding of that, and that is foundational to the work that we're doing. Also, from early work from Miles, this variance ratio, which is a ratio of the variability we see between those 4 replicates in the test, to a theoretical sampling variation, which comes from this binomial distribution. This is something that we have put a strong theoretical basis to, foundation to. Here we're showing the distribution of this ratio, which is actually quite important in our efforts to calculate tolerances and other things, which I'll talk just here in a moment. You can see one thing we see is that this ratio could actually be less than 1 quite often, which is a result that's helped us understand, the reality of the seed testing situations. And also this is a basis for the calculation of tolerances, which I'm going to show a tool that we use, which has been very useful in the seed testing area. Also, we show the exact distribution of the observed range of those normal seedling counts across the reps. A couple other quick examples of sampling of theoretical contributions that have been made. Here's an example of one from 2015 of using some Bayesian statistics, looking at taking a sample of seed and observing a result, and using that prior information to make a decision or give some information on a subsequent sample that could be taken. This is in the GMO testing area, but it could be applied more broadly in other areas, certainly. Another example in collaboration with our GMO committee colleagues within ISTA, a paper that was a white paper that was written around looking at GMO purity estimation. So for biotech trait purity estimation for a bioassay, which you need to take into account germination. So this paper looks at these paired together, looking at germination coupled with trait purity. And then finally, looking at the overdispersion of binomial data, or basically what we mean here is we get seed testing results, and some variability has to do with just the sampling of different seeds, pulling random samples of different seeds from a lot. Also, there's some other additional source of variation, maybe due to the lab and such. So here we're looking at this overdispersed data, or we're building in additional variability due to the laboratories, and this follows this beta binomial model that again is And again, all of this work is really provides that strong foundation for the development of tools and for a lot of the work and the statistical solutions that we come up with. Okay, so moving into the tools themselves that came as a result of that strong theoretical foundation. Again, this is a sampling of the tools. These tools are all available at these two web links that are from the International Seed Testing Association website, the Statistics Toolbox, as well as the Germination Toolbox. Again, this doesn't represent all of the tools, but this is a nice sampling. And most of these tools are in the form of a Microsoft Excel spreadsheet to keep it simple and to keep it available to people around the world, some of which maybe don't have the technology that some of us have. So we want to keep it in a simple vehicle, in this case, Microsoft Excel. Some of the tools are, a limited number of the tools are in R Shiny apps. So the R programming language, statistical programming language. There's been some, Jean-Louis has written some R packages for some more of the sophisticated tools that we have. That's a limited number of tools are in that type of an environment. SeedCalc and QualStat are two acceptance sampling tools that we use a lot. Got a lot of utility. This has been cited a lot over the last 20 years and used a lot around the world. There's other tools here. A tool here, for example, to test it, where we a company or organization builds a seed mixture of multiple types of seed or multiple maybe varieties of seed in a mixture. This tool will assess how well we hit the target mixture. Maybe it was a 25%, 50%, 25% mix. Well, how well do we hit that mixture? That's what this tool does. There's a graphics tool. This box plot calculator is a tool to help you visually display results. This germination tolerance and confidence interval calculator is one I'm going to highlight here in a little bit more detail, but it's a way to calculate tolerances, seed testing tolerances. I'll explain that some more. SeedCalc Stack 9 is a similar to SeedCalc. It's used when we have multiple seeds or multiple traits stacked within seed, and how you address that when you're doing seed purity testing. Likewise, also we have a heterogeneity testing calculator, which allows you to assess, if you take multiple primary samples from a lot, how you can assess the degree of heterogeneity, or the opposite of that, homogeneity within those batches. Then finally, maybe the newest addition to the tools, which isn't presently available on the ISTA website, but we will be shortly in the future, is a tool that we use for purity and other seed determination to calculate a minimum working sample weights for introduction of new species into the ISTA rules. Okay, so let's look at a little bit closer at some of these tools. First, SeedCalc. Again, this is one of the oldest, the tool that has one of the greatest longevity, I think, amongst the tools that we have offered on the ISTA website. Again, it's used in the area of biotech trait purity testing, also GMO adventitious presence and purity testing. It also has utility, though, in the seed health area, where we're testing for the presence of pathogen, in the physiology area of germination, vigor testing, and beyond. It has broad utility beyond even beyond seed testing. Has a lot of utility. It's been used even in grain testing as well. But really, the idea is in these sheets that I'll show, this one, starting with this one, is the yellow cells in these Microsoft Excel spreadsheets are inputs. Everything else are outputs. So you can enter these in, and in this particular case, we have a case, maybe this is for adventitious presence testing for unintended GMO events or traits. We're testing 6 pools of 500 seeds here. We enter those into the spreadsheet, and then we tell how many positive pools you can have and your decision to accept or reject a seed lot. So here we enter 0, so we don't allow any positive detections. And you can see here that we have a customer requirement written in here of 0.1% impurity, which the customer wants this level or below, a customer or a governmental organization. We also enter in here producer requirements as well. So for this particular testing plan of testing 6 pools of 500 seeds and not allowing any positive detections for the presence of a GMO trait event, we have a very high confidence, 95% confidence here that that lots that pass this testing plan will have a impurity below at or below 0.1%. Okay. This is specific sheet is used where we have a qualitative assay. This means an assay that that gives a binary result. Either it germinates or it does not germinate, or it has a GMO event or it does not, it has a pathogen or not. So this binary qualitative response, we would use this particular sheet. This sheet here is used where we have an assay like a real-time PCR or some type of a protein-based assay, for example, where we get a quantitative result from the assay instead of a qualitative result. It's very similar utility to the last sheet that I shared for a qualitative assay. The yellow cells are inputs. The other cells calculate risks and such around the customer's requirements, as well as around a producer's requirements for the seed. So very similar utility to the last sheet, but with a quantitative assay. Finally, within SeedCalc, there's we're only giving you a sampling of there's more than just these 3 sheets in SeedCalc as well, but we're just giving you some that are used a lot, where there's a lot of utility. Here we show that same testing plan of testing 6 pools of 500 seeds, but we have a lot here that didn't meet the requirements. You notice that we have 3 positive pools instead of the required 0. Well, we would like to enter these results into SeedCalc and get an idea of, well, what is the level of impurity in the sample, and SeedCalc will calculate that at about, in this case, about 0.14%. It also calculates an upper bound on impurity, a statistical upper bound of the impurity, upper bound of impurity in the lot that we drew the sample from. And in this case, it's about the level of impurity we're very confident is below, say, 0.4%, as we see here. Okay, so this is quite useful after you've done some testing results to enter your results in here. And there's a tool in here where you can enter them in for germination as well, or other types of tests. This is just a very short primer I've given you. We don't expect you to be able to effectively use SeedCalc. There is a reference here in the Seed Testing International from the October 2020 issue. There's a very nice article. It was written by Jean-Louis, myself, and also colleagues from the ISTA GMO Technical Committee. We wrote this article that talks of how you effectively you can use SeedCalc. There's a lot of nice examples in there, so this could be a very good reference. Also, Jean-Louis and I have taught numerous workshops on this topic around the use of SeedCalc, and we could also test those into the future if approved, as approved by ISTA. This also work was done in conjunction with the writing of Chapter 19 of the ISTA rules, as well as a GMO testing handbook, which is in development. Another tool that we use that's got a lot of utility is this germination tolerance calculator. Again, in this tool, it's an Excel spreadsheet. Again, the yellow cells are inputs, these blue cells are outputs. So in this case, we enter, we're looking at calculating a tolerance or the maximum range between test replications. So from that cartoon I shared from the 2019 paper, we had a test of 400 seeds for germination. We have 4 test reps, each of 100 seeds. If we take in this example, we take the average of those 4 replicates and get a germination of 92%. And so we have a maximum range here of 11. So across those 4 replicates, we look at the range of those, and as long as those 4 replicates are within 11 germination points of each other, then they would be within tolerance, or they're acceptable. the variability between replicates is acceptable. Now this actually comes directly from the ISTA rules. The same tolerance. If you go to this table in the ISTA rules, you'll get this exact same tolerance. So you might ask, well, how is that useful? We already have the tables. Well, this tool gives us flexibility to look at other possibilities, to use the same logic from Miles and that the International Seed Testing Association and also other groups like the Association of Official Seed Analysts in North America are using to calculate other tolerances. So here we have, we're looking at one that's not necessarily in the ISTA rules, but it's one that could be used very effectively within an organization or company. We're going to test two pools or two replicates of 50 seeds for a total of 100 seeds. We calculate the germination between those the average germination between those two replicates at 92, and again we get a tolerance that we can now use to see if those replicates are acceptable, if they're within an acceptable range of each other, just for our quality control efforts. So it shows you the flexibility. All of these, for this particular tab, these are tests that are done within one laboratory. Now we'll look at a case where we're looking at tolerances of tests conducted between laboratories. For example, a company may conduct a test for labeling and issue an ISTA orange certificate. Maybe a government organization comes back and tests that lot to ensure that the company's is labeling properly. And so we have a test done by the government agency, a test done by the company. So we have two tests done in two different laboratories. And we have this, we can use this particular sheet within the germination calculator. An example of, here we have two tests that were conducted, one at each of those different laboratories. It was a 400 seed germination test. The average germination was 95. So for example, maybe one lab calculated a 93, another one calculated a 97 for an average of 95. The range, the difference between those two is 4. The maximum allowable range is 5, so we would be within tolerance. Or those the tests from those two laboratories are within tolerance, or they are acceptable. The difference between those two is acceptable. This also is in the ISTA rules, but again, we have this tool allows you the flexibility to look at other combinations. So here we're looking at tests conducted in three different laboratories, 200 seed tests. These are all done, of course, on the same lot of seed. We get an average of 95 of the three. For example, maybe one lab got a germination calculation of 93, another at 95, the third at 97, for an average of 95. We have the maximum range of 8. And so as long as the range of those three test results are within that tolerance, then that would be acceptable. The tests across those three laboratories would be within an acceptable range. So we had a range of 4 there. We're within that maximum range of 8, and that would pass. That would be acceptable. So yeah, another example of the flexibility of this tool. There's a third sheet within that tool where you can, similar to what we did in SeedCalc, you can enter the number of seeds you test, enter a confidence level, in this case a 95% statistical confidence, the calculated germination in the sample, the percentage of normal seedlings, and then we get a reported germination as an output, as well as a two-sided confidence interval. So we observed 94.25% germination in the sample. You can see that within the lot, we can make a statement that the germination is in the range of, from that particular seed lot, it's in the range of 91 to 96%. Again, yellow cells are input and the blue cells are output here. So finally, one of the newest tools in our family of tools that we developed is this tool that's being used for purity and other seed determination minimum working sample weights. So within the rules, we need to have a purity sample of 2,500 seeds, or at least 2,500 seeds. and for other seed determination, it'd be 10 times that, or 25,000 seeds. So this provides a nice tool for our purity committee, and for the introduction of new species to calculate, considering multiple sources of variation, including variability between varieties, variability between lots, variability between labs doing the testing, of course, variability between the within the lab, the seed samples themselves. It considers all these sources of variation to give a conservative estimate of a minimum weight to achieve the 2,500 seed limit for purity and the 25,000 seed limit for other seed determination. The tool gives flexibility. Sorry, this is a little bit small, but to where we can conduct this study across multiple labs for where each lab looks at distinctly different seed lots from the other labs, or situations similar to our method validation design where each laboratory evaluates the same seed lots. And so if there's some difficulty in movement of seed, we may not be able to get all the seed lots to all the labs. This gives you the flexibility to use either approach. Okay, the results on 100 seed samples, 100 seed sample weights are entered into this spreadsheet for each of the laboratories. And that's the cells in yellow. You can fill out the other cells in yellow, and then it will do all these calculations. It will fit these relatively sophisticated statistical linear mixed models, but it does it all within this Excel spreadsheet to get that estimate, to get a conservative estimate of for the minimum seed weight to meet those seed counts that we need for purity and other seed determination. Here we show an example with some data where we're entering in these 100 seed weights in grams. And then over here, we're showing in this particular case, we have 7 laboratories involved, 5 seed lots for each of those laboratories. We'll get some flags in here if we're not meeting some of the guidance from the purity committee and the rules. And we calculate these variance components, and then we use those to again get that conservative estimate of a seed weight to meet the purity requirement and the other seed determination requirement. And another nice thing about this tool is that it will generate the results will light up as red if there is an outlier here, and we're using this method called Grubb's method to calculate outliers. Jean and I built that in there, and so these can actually be suppressed or taken out, these outliers, and to do these calculations. The calculator also handles very well some imbalance. So I'm going to share. I'm going to do something here. Try something here. I think I have a couple more minutes, Andreas, if my clock is right. I'm going to share a kind of a real example here. So this is that latest tool we've added to our collection. Here we have these results. I'm going to show you if I enter this result here as 6 grams, you'll see it will light up as red, showing that identifying a Grubb's test outlier. So those can be suppressed. This tool also allows for imbalance. So if you have a different number of reps for these different seed lots, it can account for that imbalance. All right. So this is what Jean-Louis and I have to share with the group here today, and we appreciate your attention. We really look forward to, with a strong theoretical foundation as our basis, we look forward to developing other tools and running other statistical analysis and calculations for our colleagues within ISTA, but also some beyond that as well. So thank you for your attention.
Thank you very much, Kirk. We still have time for a few questions. You were well in your time. Thank you for that. There's one question: are there tolerance tables available for seed health test results?
Well, that's a really good question. I think the answer is no. Maybe I'm not. Maybe Andreas or someone from the secretary may have a better answer. But I believe the tolerances today are available only for germination today. Yeah.
Yeah, that's what I know as well. And is it necessary to do a heterogeneity test routinely for every sample operation?
That's a question. Yeah.
Yes.
Okay. I think we're running that heterogeneity test. I think as part of the ISTA audit, the lab accreditation ISTA audit. There's a requirement there to assess our processes. Maybe not on every single seed lot, but we need to show on the processes that we have homogeneity in our seed lots. So the answer is no, not on every seed lot, but certainly for our ISTA accreditation, for the audits that ISTA does, this is part of the requirement.
And how do we handle situations where the number of replicates and or the number of seed tested are different between laboratories involved in the comparison?
Yeah, that's a really good question.
I've got only good questions here.
Yeah, right. Yeah, all great questions. This is something that yeah, theoretically we could handle. This is something that Jean-Louis and I could consider. I would say, you know, within this calculator we're using today, we're assuming this balance or similar numbers of seeds per rep. But in theory, yes, we could do this. It's not, the flexibility is not available in the tool today.
Mm-hmm. And the last question here: is there a way to view the formulas, VBA macros in the tolerance calculators or even SeedCalc 8 to visualize the calculations?
So today on all of these calculators, we protect the sheets to protect the users, you know, and but there is for those who have specific interest, you can, like in SeedCalc, it's not password protected. You can go into SeedCalc and then protect the sheet, and you can actually look at formulas. You can look at the Visual Basic code. You're right, it is Visual Basic code that we're using in these applications. For the tolerance for this particular germination calc I'm showing on the screen, it is password protected, but we could, getting a hold of Jean-Louis or myself, for specific cases, we could give you privilege to, you know, to look at those formulas.
Thank you very much, Kirk. That's all the questions I have. So no need to answer later on. There's another remark coming in saying that regular ISTA seed test results are based on the rules. So to have uniformity across labs, tools allow more flexibility, research, and other studies. So that is an answer from the back. Kirk,
Good.
if you could give David the presentator mode, and I will introduce David shortly. David Merritt is a principal research scientist of the Department of Biodiversity Conservation and Attraction based at Kings Park and Botanic Gardens in Perth, Western Australia. His research interests include fundamental and applied aspects of biology, physiology, and ecology of seed, including studies of seed storage, behavior, dormancy, and germination. His work focuses on supporting the ex situ conservation of plant biodiversity through seed banking and on developing techniques for the propagation and use of seed for ecological restoration and threatened species translocations. David, the floor is yours for the next 30 minutes. Please go ahead. Thank you very much. And don't forget to put up questions in the question and answer box. Thank you.
Okay, thank you very much. Hopefully everyone can see my screen here.
Yes.
Yeah, we're good. So thanks for the opportunity to talk. My talk's a little different. I'm going to talk about some experimental data, some of the approaches that we're taking in Western Australia to study seed traits and how they can inform restoration practice in the diverse landscapes that we have here in Western Australia. This is obviously a vast topic. There's many groups around the world doing some great work in this area. This slide here shows I guess a non-exhaustive list of seed traits that are involved in defining the regeneration niche of a species. So you could view that as the specific abiotic factors or conditions that a seed requires to germinate and the seedling to emerge and survive. Today I'll talk a little bit about dormancy and germination, emergence depth, and also how they interact with some of these soil temperatures, the rainfall conditions, and also substrate properties. And what we're trying to do really in restoration is define this establishment niche for the seeds of different species, and then to improve our practice and improve the proportion of seeds that go forth and establish, we're seeking to modify that niche either by modifying the seed and the seed properties or the substrate properties. So I'm going to focus on a few examples, mostly from the northwest of Western Australia. So as shown in the map up here, this is the Pilbara region of Western Australia. It's a semi-arid grassland. You can see from the picture there, it's dominated by a particular genus, Triadia, or we call it spinifex grasses. This is a pristine area for the most part. It's a beautiful landscape, as you can see, but it's also heavily impacted by mining, and much of the context of the research we do in our labs is to look at how we can improve restoration after mining. In the Pilbara region, a lot of the mining is iron ore mining. So this slide here shows basically the restoration landscape that we're left with once they have completed the extraction process. So iron ore mining is pit mining, and it creates larger amounts of this waste material. They dig large holes, so many of the large mines can be more than 1 to 2 kilometres deep, several kilometres long and wide, and so we get left with these really vast areas of waste rock material that are mounded to resemble somewhat the surrounding landscape. But you can see really it's quite a hostile looking environment. Most of these slopes are tens to hundreds of hectares in size, and they're completely devoid of vegetation. So at this kind of scale and in this environment, really we're entirely reliant on seeds to return biodiverse plant communities, and that is direct seeding via machinery. That's really the only viable means in which we can get plants back at this kind of scale. So this is why we focus so much on seeds and seed technology and how we can improve their establishment and waste in these waste materials. And I should add that, as Simon alluded to in his talk, our current establishment rates with current practice can be very low. So it's certainly less than 10% of seeds sown to these sites would establish, and commonly it's 1 to 2% or even close to 0%. So we certainly have some improvements to make. All of the seed is wild collected in this region too, so we want to be able to use the seed much more effectively than losing 98, 99% of it, so that we can reduce the pressures of wild seed collecting from the surrounding landscape. So one of the approaches that we took in this region to study seed traits and how they interact with the environment is to build this rainout shelter. So this is at one of our industry partners' mine sites. It's the Mount Wellbat mine site in the Pilbara. And we built this rainout shelter so that we could study seeds in a more natural kind of environment. So our laboratory is in the southwest of Western Australia, and we can do, and indeed we certainly do lots of experiments in the incubators, etc., but it doesn't really replicate the environmental conditions up north in the Pilbara, where the temperatures are very hot. So summer temperatures are 35, 40, even up to 45 degrees. It's a summer rainfall zone, so that's the growing season, and it's as I said, a semi-arid landscape, but it's a cyclonic-driven system too. So that means that the rainfall is sporadic and variable in time and space. So if we were to do field experiments and just rely on natural rainfall, there's a good chance that we could go a number of years even without any significant rainfall, and we're unable to test our seed treatments in any reasonable time period. So part of the reason we built this rainout shelter was also to risk manage that research process so that we could get some results in a reasonable period of time. So it's simply a covered structure. You can see here we have 32 soil plots of 4 by 4 meters, and they contain different soil substrates. So some natural topsoil, which is the most desirable substrate for restoration, but it's in very limited supply. And then we had a number of different waste types, which is what they create the waste dumps from. So I'll run through an experiment that we did initially in this rainout shelter, and the concept here we're really interested in is these early life stages of plant establishment. So some of the work done by our colleagues in the US were I guess the first to start to really unpack these early life history stages, and so you have your seeds. They need to first germinate underground. Those seedlings then need to emerge from the soil into the establishment phase. So you've got a number of steps in the process, and some work by Jeremy James in the arid systems of the US back in 2011. He found, his group found that in their systems, the probability of seeds sown to germinate underground was actually reasonably high. You can see an example there of 50 to 80%, but then they found that this emergence phase, so the emerging of the germinated seedlings, the probability of that happening was only 1 to 5%, but then of those seedlings that did emerge, probability of establishment was reasonably high. So similar work by Julie Larson with some figures down there as well, and Julie's work also showed with a much larger number of species, 47 grasses, that the probability of emergence was in fact a strong predictor of plant establishment. So this kind of work really set us up to think about, well, what's happening in our systems, and where do we think the kind of failure points are in these first stages of seedling establishment. So the experiment I'll outline now was basically we used the rainout shelter to understand how different substrate types and water availability influence seedling recruitment, and we're really interested in modelling the probability of these different phases, these life history phases of germination, seedling emergence, and early survival, so that we could define those stages we felt were most limiting, and then look to how we might be able to use different seed treatments or soil management techniques to address those limitations. So we used 5 species in the initial studies. This just outlines our experimental design. So we had 2 species of acacia and an indigofera. These are all legume species, they're shrubs. And then 2 of our spinifex species, the Triodia, Weisiana, and pungens. We did treat these seeds for dormancy before we did the experiment, so I'll come back to dormancy in a moment, but really to get any sort of emergence in these systems with our seeds, we need to manage dormancy. So we treated the seeds first, and then we focused on different rainfall regimes, and we were particularly interested in trying to identify a minimum threshold of rainfall that would be sufficient to initiate germination and establishment. And so you can see the different regimes we had there from, and we just watered over the first 7 days, and then no longer for the rest of the experiment. We had one regime that was very high, which we deemed to be non-limiting, so that gave us a little bit of a comparison of performance under the lesser rainfall regimes. And we monitored these different life stages, germination through to early survival, and in fact looked at biomass too. So what did we find? Well, there's some data here. I've got two panels we'll talk through. On the left-hand side, just presents basically the raw data, if you like, the cumulative fate of 1,000 sown seeds. So this data is normalized just so we can compare between the different species. And I've just got data for 3 species because the 2 acacias and the 2 triadias behave relatively similarly. So this left-hand panel, what is evident, we have our maximum germination in the laboratory here, so under optimal conditions. So you can see that acacia actually germinated relatively well in the soil in comparison to the laboratory. We didn't get anywhere near that germination in Indigofera. Triadia was also germinated quite well. I think also what's probably most obvious in this left-hand panel is the 80mm rainfall. So our lowest regime did certainly impact on the ability of the seeds to germinate, really across all of the different soil types that we've got indicated in these different colours. So it does look like the 80mm is insufficient for germination, but then the rest of our rainfall regimes from 120mm up to 280, they were broadly similar. On the right-hand side here, though, this is probably more easy to interpret when we start to think about which life stages are most limiting for the different species. So this is the data showing us the probability of these seeds moving through these different life stages of germination, emergence, and then survival, in this case to week 6. And so what is hopefully evident to you all is that, in fact, the probability of seeds emerging was actually very high. So we really find that germination for at least the Indigofera and our 2 Triadia species, germination, the probability was quite low, and that was the limiting life stage. Whereas the probability of emergence, in fact, was quite high. So this is a little different to the data that's been found in the US, where they found that this emergence phase was, the limiting factor. I guess the other point to make here is that we have species-dependent limiting life stages, if you like. So germination, certainly for the Triadia, for the Indigofera, it's probably germination and survival. Acacia, germination emergence is good, and it's probably survival, which is the limiting stage. And the other point just to make is the substrate properties. So this waste type in particular, seedlings really struggled to survive. You can see the probability of survival is really quite low in this waste type relative to, for example, the topsoil. We did have some blends, so we blended the topsoil and the waste in a 50-50 mix, and you can see that has some capacity to reinstate or support survival, so particularly evident, for example, in the acacia. So between the pure waste and the blend, you can see there's quite a difference there in survival. So I'd like to just focus a little bit on dormancy now, just touch on that. So as I mentioned, we have really this germination phase for us remains to be one of the key limiting factors, and we see this across many experiments in different systems. And when we think about the natural seed life cycle, so seeds are shed at maturity, and they'll enter the soil seed bank, and I guess a simplified kind of way of thinking about it is most of these seeds are dormant. They'll have a physiological component of dormancy. Some of them will have physical dormancy, some will be non-dormant, but in our systems, the majority have some type of dormancy. They may cycle back and forward between the dormant and non-dormant state, waiting for appropriate conditions to germinate. They need to persist in the soil as they're waiting for those appropriate conditions to germinate. Some will be lost to aging or predation, or even erosion and removed from site in context of restoration. And then they'll germinate if the conditions are right. So this is kind of the natural seed life cycle, with dormancy being one of the key factors controlling whether we get to this germination phase or not. In our restoration practices, well, we actually collect our seeds before dispersal. We then store them in artificial conditions for, as far as the seed is concerned, an arbitrary period of time. We come along and we sow them back into the source seed bank, and we expect them really to germinate at the first opportunity, so at the commencement of the growing season. So we are kind of expecting that they're going to bypass all of these natural processes somehow, even though they're not exposed to the natural conditions. So of course, what we actually have to do is manage the seeds through this phase pre-sowing, and think about pre-treating them to break their dormancy so that they're able to move into this germination phase at the earliest opportunity, so when we get sufficient rainfall. There is some interaction though with persistence and dormancy, and some research I won't cover today, but just to say that we do know for some species, the Tradia, for example, when we render them non-dormant, they don't persist in the soil for as long. So that does mean that if we don't actually get suitable conditions for germination after we've sown them, then we might lose them. So I think there is perhaps a case to think about adding a blend of seeds, perhaps 50% that are dormant, 50% that are non-dormant, to add in a kind of bed hedging capacity. We're doing a little bit of work to pursue that. Because we get a lot of questions about why don't you just sow the seeds in the soil and let nature take its course. But these periods of dormancy break can be quite long. We have examples of seeds with physical dormancy. 5 years after we place them in the soil, they're still dormant. We have many examples in our system of species that produce seeds that need smoke to germinate, so they're not going to get that exposure in a restoration context. So we certainly need to treat them for dormancy, but I think it is worth thinking about whether we can build in this kind of bed hedging capacity and have a mix of dormant and non-dormant seeds at sowing time. So to be able to achieve all of these things and manage dormancy, we need pre-treatments that are reliable and repeatable and applicable to large quantities of seeds. So as I say, we collect our seeds, we store them in artificial environments. Most of them will be dormant. We want to take them out. It might be after a few months or many months or even a few years, and render them non-dormant at the time we're getting ready to sow them. So this is fairly easy for some types of seeds and some types of dormancy. So physical dormancy, for example, this is an acacia, so they're relatively easy to manage to alleviate dormancy. But many of our species with physiological dormancy, and in the pilbara, many of the grasses, they're quite challenging to break dormancy. So that's why we focus a lot on that. And we use the Baskin system quite a lot. This is some data from just a recent review paper where we had a look at how we were going at breaking dormancy in different regions of Western Australia. So the map up here on the right kind of shows the regions in which we work, and we broke down data on seed dormancy from species in these different regions. So this left-hand panel is just simply an example of the proportion of species that have the different dormancy types recognised under the Baskin system. So that is academically interesting and ecologically interesting to think about, the proportion of seeds that might have physical dormancy or physiological dormancy. And for example, we don't see many seeds with the morphological components of dormancy in regions except for the southwest of Western Australia, which is where we have the cool temperate climates. But then on the right-hand panel, this is where we analysed the proportion of species with the different dormancy types in which we were able to achieve greater than 75% germination. So we like to use that kind of threshold of 75% germination to say that we're happy that we can break dormancy. That's a somewhat arbitrary proportion or percentage, but we feel it's reasonable. And I think it allows us to, for example, recognise that certain dormancy types, like those with the morphological or physiological components of dormancy, are usually a little bit more challenging. As I said before, physical dormancy is fairly easy to manage. And so I think one of the benefits of the Baskin system is that you can identify the type of dormancy of the seeds without necessarily knowing how to germinate them in the initial phases, and that can also help us then to direct our research resources. So we know that we need to spend more time and more effort on species with these physiological, or morphophysiological dormancy. So dormancy, of course, is not the only part of the germination journey, if you like. Even non-dormant seeds require appropriate conditions to germinate, and this graph here just shows a little bit of an example of where we study other seed germination traits. So in this case, thinking about germination temperatures and base water potentials. And so what we're interested in here is being able to, I guess, predict when seeds are going to germinate, and look at this graph shows different substrate properties. So this species grows in these banded ironstones here. This, in fact, is an example of a threatened species. And on the left-hand panel here, this is some data from a natural system, and then some data from one of our waste rock systems. So the top two panels is simply data logger data measuring soil temperature and soil water potential. These seeds like to germinate around 15 to 20 degrees. 25 degrees is about their upper limit. So this green panel I've got here just shows the growing season. So from May to September in this region, when the temperatures are suitable. And then what we've got in these lower panels with these dashed lines here is the cumulative time at which the soil water potential was sufficient to support germination. So based on the base water potential of the seeds. And there's two values here. So this value here is for smoke treated seeds. So when we treat these seeds with smoke, they're able to germinate at lower water potential. So that's a useful pre-treatment. But this data shows that even for our smoke treated seeds, the cumulative time at which there was sufficient soil moisture for these seeds to germinate was only about 500 hours over the entire growing season. So that's about 3 weeks, and then in the waste rock it was even less. It was less than 400 hours because the waste rock had lower soil water holding capacity. So by gathering this type of trait data, we're able to, I guess, more finely define the germination niche and understand what and where the opportunities might be for the seeds to germinate, and also evaluate our pre-treatments and whether they're in fact widening the conditions that may be suitable for germination, and then giving them a better chance to germinate and emerge. So in the final part of my talk, I just wanted to talk a little bit about our work on direct seeding. So I mentioned in the beginning that we need to use direct seeding machinery on these waste rock dumps, and we kind of have a couple of options to improve the establishment rates, and that would be just put more seeds out there, or we can try to increase the number of microsites through modifying the substrates. And we will eventually get to the point of diminishing returns, and a point where putting out more seeds will have no continual effect. And what we want to be able to do is again use our trait data to modify the seeds and increase these microsites that are suitable. To create a seed mix, ideally we're talking about, well, we're thinking about our desired plant community we want restored based on our targets and the limitations of the substrate. That should then lead to us thinking about the number and proportion of seeds and species in the mix, and ideally we would be able to adjust our seeding rates of different species based on the likely establishment rate. And this is where we're quite data deficient and where all our seed trait research comes in. So the current practice for direct seeding in these environments is to use very large bulldozers. That's what they have on site. That's what they use for the earthworks. They're 11-ton dozers. They're not really suited to direct seeding, but that's the powertrain they use. And then they tend to just use a simple hopper system and broadcast the seeds onto the surface. So we know surface sowing is bad, and so a lot of our research with our engineering colleagues at the University of Western Australia has been looking at how we can design direct seeding machinery that can more precisely sow seeds in these sloped and rocky landscapes. And we're particularly interested, I guess, in sowing depth. There's a little bit of data here from a study we did, again, in the rainout shelter with our Troidia seeds, where we were trying to understand optimal sowing depth. So this graph basically shows soil substrates of increasing rock content. So there's data for soil that was sieved to different sizes, so less rock to more rock. This purple bar here is basically untreated seeds sown on the surface, which was current practice. So you can see the very low emergence in that case. What this data really tells us, though, is for our treated seed that an optimal sowing depth is somewhere around 5 to 10 millimeters, and that increasing rock content affects emergence, and it becomes increasingly problematic as sowing depth increases. So we really need to be sowing these seeds not on the surface, about 5 mil below the surface, but we also don't want a lot of rock on the surface. And so this kind of precision is what the challenge is. So it's much shallower than agricultural seeders, and again we've got the sloped and rocky landscapes, so we need to be able to develop machinery that can have this level of precision for our native species. And then I thought I'd just touch on the delivery process. So many of our seeds, our Troidias, the grasses, have these hairs and appendages which are problematic for seeding, because in fact the seeds just won't even go through the hoppers. They bridge and block the hoppers. And also, if you want to apply some seed enhancements such as seed coating, you can see in the raw state we can't apply those coats. So we developed this seed flaming technique. I'll just play the video quickly here. It's basically a rotating drum, and you can see the seeds spinning there. They're exposed to a flame. We can change the number of flames and the size of flames. And basically what this does is removes these surface appendages. So this is the raw state, and this is the flame state. You can see these are much smoother. We can coat them. This data here on the right shows how the seed volume or the seed mass is reduced with flaming time over about 15 minutes. So this not only helps us with seed geometry and coating, but it reduces the volume required for storage. The main reason, though, we want to do it is to improve seed flow through the hoppers, and so we really needed to come up with a way of measuring seed flowability. So I'm not sure this is a functional seed trait, but it's certainly a seed trait that impacts our seeding practices. So some work by Elvin Ling during his masters. He took some inspiration from the bulk solids or the powder industry, and so they use this technique called the angle of repose, and so basically created a hopper. The seeds pass through, and as they fall to the base here, it's a simple measure of this angle here. It's called the angle of repose, and that's a measure of the cohesiveness of the material. So there's a range of species here in this work. You can see across the top the unflamed and the flamed seeds, and then the angle of repose. So basically flaming for different periods for all species is able to improve their flowability. So this was a nice way of us quantifying seed flow and helping us to develop flaming protocols. So that would be the time at which the seeds of the different species are flamed, and whether we use a large or a small flame, and 1 and 2 flames, etc. Of course, we don't want to impact on germination, so we need to be careful about that, and we do a lot of work around the germination of flame seeds. And what we actually find for many species is that germination is either not affected, provided we don't flame them for too long, of course, or we can get a good improvement in germination. So this is germination of the seeds in their natural dispersal units, as we see up here. Often these covering structures impose dormancy, so presumably the flaming weakens those covering structures a little and helps to alleviate dormancy. And you can see that for many species, in fact, germination is increased. But again, just as per dormancy and per germination traits, etc., we see that there are species-specific requirements for flaming. So this does require a little bit of protocol development. You can damage the seeds by flaming them too long. Some are very susceptible, but we're able to moderate that through modifying our flaming processes, including through using water and softening the flame by injecting water during the flaming process. So just to wrap up my talk, I just an example here of some actual field data showing that we can get an improvement by putting all of this together. So this is a 5-hectare trial up in the Pilbara using direct seeding machinery that's designed for more precise seeding. And on the left here, we had our untreated florets of 2 Troidia species. And you can see even when we sow them precisely, if we don't treat the seeds for dormancy, we get poor emergence. But once we do treat them, and there's a range of different treatments we tried here, and in fact, most of them gave a similar result, and that is that we get a 5 to 6-fold increase in establishment by sowing the seeds precisely and treating them for dormancy. And you do have to have a little bit of luck. This site was sown and then a cyclonic system passed over it about 3 weeks after sowing, dumped about 150 millimetres of rainfall, so it crossed that 80 millimetre threshold I was talking about, and that was sufficient to initiate the establishment that you see on the right. And this is one of the better results that we have, and we're able to do this, as I say, on a 5-hectare scale, so we're starting to scale this up. So I'll finish it there just with a summary slide, just to get you thinking again about seeds. They're complex, they're finely tuned to the environment, they possess this suite of functional traits influencing their behaviour. For ecological restoration, we need to understand those traits and seek to modify them to improve their chances of emerging and establishing, and that is the way that we'll be able to achieve our goal of these biodiverse restored landscapes. Thank you very much. Of course, I'd just like to acknowledge that there's many people and government and industry sponsors of this work, and these are the people contributed to the data I presented today. So thanks for listening, and happy to take any questions.
Thank you very much, David. That was a very interesting presentation, and I got one information on a question here. Have you tried covering the seed with hydro milk or milk material?
Mulch. Yeah, we have not tried that approach. That approach has been taken. It's on some sites, and they also use it on the edges of roads, for example. We have been more focused on the seed and the seed technology, as I outlined, rather than hydromulching.
Good. That was actually the only question I have.
Mm-hmm.
Thank you very much to all the speakers, to the nice introduction we got on INSR from Kingsley. We had a good session from Simone Pedrini. There were a number of questions which are unanswered, and Simone will try to do that later on. And Kirk was having a great presentation and with a lot of questions around the ISTA tools. And during the session, I also shared the link to the ISTA website where all these statistical tools could be found. So that is very easy. You go on the ISTA website under tools and go to the statistics committee, and you will find all the tools delivered by the statistics committee. And there are other committees as well who can deliver tools for other purposes. And David, thank you very much for your presentation as well. I would like to end just with where we started in the session, with a short introduction, and I hope you can see my screen now.
Yes.
Just to remind you that these events are upcoming, the ISTA annual meeting. You find it also on the ISTA website, of course, the ISSSB annual conference, and also the SER 2013 World Conference on Ecological Restoration. I thank everybody very much, and would remind all the people here that if you want to have a look at this session again, this webinar, you will find it on the ISTA YouTube channel during the next days. Thank you very much, and have a good evening or whatever, wherever you are. Thanks.